messenger leaving the nucleus and going out and finding a ribosome, inserting itself in the ribosome, and the ribosome reading it and translating it into a protein. And that's what this whole, we're right here though for transcription. We haven't gotten here yet, but let you know what's going to happen. He's going to leave the nucleus. Because right there is the boss. That blue thing is the boss. But he can't leave. He can't get through the hole right there. So he makes him a messenger, and he leaves, carrying with him the message from the boss. It works really well. Now you said we're going to start 16. What was that? Oh yeah, y'all gave me that already. There's no 16. Are we on translation?
I said slide
No, transcription.
15. The slide 15. Not the
Oh, you mean this?
Yeah.
Oh, I got you. Good. I was looking for right there 15. Okay. Let me look. Now, we saw this one, correct? Or not correct?
We saw 15 even.
Okay. Let me start there and kind of overlap a little bit. And this is not directly. Let me go back to my opening. You see right here? There's the instructions. But this one does not talk to the thing that makes the protein. So indirectly, this is what has to happen. This is your indirect route. And if he gets the message wrong, if I whisper to you a secret, and you whisper to Hillary, and when it gets to her, might it be changed a little bit?
Yeah, because you can never repeat somebody.
Exactly. Well, that's the thing about this. This has the same potential to mess up. Because if I whisper you the secret, and you tell Hillary, by the time you get to her, it might be changed.
Mm-hmm.
But the way this works, do you know what binary code is for computers? 1 and 0, 1 and 0. It's all binary code. Everything's is all binary code. Now, I'm not a code writer. Now, I did take a class in college one time where you had a key punch cards. And you line them up and you put them in a file. And that thing went to the computer. And it could be the simplest thing where you said, if the answer is Columbus, then say yes. If the answer is no, say try again. And the question might be, what school do you go to? And then the response would be Hardaway. Well, then the computer say no, and make you try again. And when we did it, it was called passcode. And I'm thinking to myself, I am not going to be a computer programmer. But then when these came out, like, I guess one of the first ones out was Microsoft stuff. You ain't got to write your own code. And it's like, took off. That man's a multi. So if you find yourself a product that folks can't live without, you got to make it. Mm-hmm. And like the computer right now, if they said no more computers, I'd quit teaching. I'd start planting gardens because I need it so much. But anyway, this is your indirect route where he tells this thing, and this thing tells this thing, and this thing, the ribosome, actually makes the protein. So that's what we're getting at when I said indirect route. Okay, so there's your indirect route. When I say indirectly, I mean that the DNA never really talks to the ribosomes. He talks to the messenger, and then he's trusting the messenger to get the job right. Okay, now there's something here that was you know that when I say C, you say G. When I say G, you say C. See, it ain't like they're a combination. Like if I said C, well, you could say A, B, C, or D. That would mess things up, wouldn't it? It's a 1-to-1, so it's hard to mess that message. If I am C, and I say C, and you say G, and she reads back C, hard to mess that up, isn't it? I mean, it's hard to mess that up. But sometimes, though, I'm here and I'm saying C, I get hit by UV radiation, and my C changed to G. It's supposed to be a G. Well, you say a G, what do you say?
C.
C. And then she says, and the protein manufacturer might be one that causes breast cancer. That's how the sunlight can mess you up, because it can actually change the sequence. And if you're going to email me, you know right now that my name is T-E-F-F-T. And if you spell it T-E-F-T, I'll never get your message, and this is just that way. It don't assume there's another F there. It just says, But the thing about it is, though, polymerase is so good, what's it mean to proofread your work before you give it to me?
Review it?
Well, the polymerase reviews and make sure now. Let's make sure this thing's right now before it sends it out here. So DNA reviews its own work. So it's rare that you have this happen, but we know it does. But look at all of us in here, all the folks in the world. Everything works fine with them. And think about all the things that go wrong. It's amazing we have our good health at all, really. Okay. There's 3 main steps to how this whole thing is done, the thing called transcription. And I'm talking about making the messenger RNA, where the DNA actually makes it. Now, first is the polymerase. DNA has two strands, right? I just use one side of it to make this. And I'm going to use my paint in a minute to show you this. Okay, so transcribe only happens when DNA is used to make a copy of RNA. That's all there is, transcription. And there's a nucleotide that tells it where to stop and where to end. Even a telegram you send says stop after every sentence. Well, the area that the polymerase attaches to first is going to promote the rest of the messenger. That's called the promoter region. That's where it can't go to anywhere. I mean, if you're going to do an email, you can't stop by saying EFFT. You got to know where to start. So this is how you start it. Now, DNA is all twisted up, right? But it's got to untwist and become 2 straight parallel strands. Like when you see it, it's just all twisted up, like a railroad track that you twist and twist and twist. Well, part of the transcription process is that you untwist it, and RNA polymerase does that. Now, you only make a single strand, though. RNA is on one strand, because 2 can't get out. Only one strand is skinny enough to get out those holes in a nuclear membrane. And it mentioned right here, the 2-stranded, only 1/2 of it makes the messenger, not both sides, just 1/2 of it. And the beauty of it is that with DNA, if one side gets damaged through UV radiation, the other side that's not damaged can pull away and then replace the correct sequence. And then this sequence over here that's messed up will be kind of cordoned off and never used again. It stays there. And those are called non-coding regions. And we know now that DNA is filled with your genes. Your genes are on DNA. And that when the genes get messed up, whether through a virus attack or a bacterial attack or the sun UV, that section can be quarantined and a new section made based on the other section. So if this section says T, then you can put A over here. There's A over here, But if this thing messes up, then you can rebuild it. And next thing you know, you have yourself a complete strand. And you still have over here the non-coding strand that was messed up. Can't get rid of it. But you ain't got to use it either. And that's why we talk about the 2-stranded DNA being the code to make a single strand of RNA. So let me say steps. Okay, first step, you got to bind to DNA. You got to bind to it. Second step, you got to elongate. You got to unwind the DNA molecule. And this is more the second step. Now I got a space. I'm going to cover that space up right there. All right. Now, this is what's going to be, at first for me, was confusing. The side you're going to use to make your RNA is the antisense side. And the side you don't use of the DNA is the sense side. That made no sense to me. Because I would think the side you would use would be the sense side. But what happens though with the discovery that when they come in and make the DNAs, that becomes the sense of the message, the RNAs. Because you got to think about it. Well, I'll show you later on. The RNA you make is just like the side that you pulled off the DNA. Because if this says A, RNA is going to be a U. But on DNA it's going to be a T. But T's equal U's. And if this side says C, then on RNA it's a G. But it's also a G on DNA. You're going to find out that the RNA message is just like the other side, except for there are no T's in it. And I'll show you later on what that means. But the thing about it is, though, the side I'm looking at to get my messenger is the antisense side. The messenger is the sense side, which equals the other DNA strip. And I'll show you, I'll do it for you in a minute. All right. This you know, I think you know, but I want to be sure. RNA has these bases. Well, DNA has the same bases. Well, if I could change that to make them line up perfect. I would like them, you know what I'm trying to tell you? I would like them, and this one only in DNA. So I'll just change the outline, haven't I? But I think that makes more sense to read it that way. If I remember, I'll change the outline on for next semester. Those kids will see it also. This is 618.
Actually, you've made a couple of changes to this PowerPoint from the one that's posted
Oh, you know,
for us.
yeah, I have. But this is a major one here. Are all this major?
Yeah, it's a lot
It's
different.
very different.
Oh, it is?
Yeah, from the one we have.
I'm going to have to redo it then. Okay, I'll just say redo the outline to match. Yeah, you're right. I've changed it all along, haven't I? I'm making it better because you're the first one to see it. And I wanted to make more sense to y'all. So I'll just redo the whole outline. But right now, I think this is a better presentation of what I put on that outline. Because it shows you G and G in both of them, C and C in both of them, A and A, and there's your only difference. And then, and I'm going to tell you, and you know that C always hooks to G, or vice versa. And you know that you have A in both of them, but the problem is, if you're making RNA, you must pair the A to the U. If you're making DNA, you must pair it to the T. Does that make sense so far?
Mm-hmm.
Okay, good. Now, step 3 is termination. How do you know when it's finished and the messenger can now leave? You had that promoter sequence, remember the one that started the whole process? Right there, it started. He starts the process. And when it's over, there's one called the terminator sequence, and he ends it. Sequence, is that thing highlighted both times? No, region, okay. Terminator region, terminator sequence. That tells the messenger it's done, take off, go to the ribosome. Okay? Now, the polymerase that's been making this, let's go with it. And then the messenger can go out into the cytoplasm. And there find the ribosome that's going to actually read the code and make the protein. Okay? Now this is continued. Now, proteins are not made by the nucleus. We thought they were for the longest time. We thought the nucleus made all this stuff. But then we realized that no, there's nothing being all the nucleus is an information center. It's just a vault of information. And so it's in the cytoplasm. So in the cytoplasm, you find the factory to make the protein, that ribosome. So the messenger must go out there to find the factory. Now, the messenger can cross that membrane because he is small. He's a single strand. If he were a double strand, he couldn't get out either because he'd be too big. Now, when you get outside there, and now you're in the cytoplasm, you're going to be attaching yourself to a ribosome. But another kind of RNA is going to bring in the acids that you call for. Going to transfer the acid to you. So we call that transfer RNA, tRNA. And then in the ribosome, as the transfer brings the acids in, it hooks them together one at a time, and slowly makes the protein. If the protein contains 40 acids, it's going to take 40 tRNAs to bring the 40 acids in. And I got a picture somewhere of that. Oh, these are some good things here. I want to stop for a moment and show you on the paint what's going on here. I've done it pretty well with other classes. I've never tried this before. So I'm going to draw this side and say A, T, C, G, let's do a T, let's do an A.
I was about to do the same thing yesterday, Mr. Chairman.
Huh?
I was going to do the same thing yesterday, but I caught myself.
That's all right. 1, 2, 3. Every 3 is a message. So I need 1, 2, 3. I need 2 more. Because the message comes in a series of 3. All right. Those 3 are a message. These 3 are a message. These 3 are a message. We call those codons. C, O, D, O, N, S. So in this strip, there's 3 codons. So every codon is 3 bases. Now, if this were DNA, and it is DNA, of course, what's going to be the matching basis? We're making DNA now. If this is a DNA molecule, what are you going to find opposite A over here? Huh?
T.
No, not DNA now.
T.
We're making DNA.
T.
Oh,
I can use a
okay.
T. I can use a T. All right. Opposite this T would be an A.
A.
And opposite this one
G.
would be G.
There's not 6.
And opposite this C would be a.
C.
I mean, this G would be a.
C.
And then opposite this T would be an A.
Yeah.
Because I'm making DNA. So I can use a T.
T.
And I can use a C.
A G.
And I can use a T. And an A. That's what the DNA looks like. Double strand, right?
Mm-hmm.
Now, let's pretend that this side is the antisense side. I'm going to read that side. All right, so I'm going to do this. I love this. I'm going to take this and see if I can't get it separated. That's going to be a problem. I want to pull this side away because it really separates like that. Now, I'm not using this side. I'm using this side to make RNA. Now, when you come back with RNA, that's when you start slapping the U's on and never the T's. Everything else is fine. Now, this is the antisense side. Now, I'm going to make the messenger right here. Now you got to watch out, like Katrina said. You're making RNA now. So what goes there now?
U.
The U does. Now, T matches with a, not a U, A, because if you said a U, that's like saying C to C. They're the same thing. U's and T's are the same thing. You can't do G to G, can you? Well, you can't do U to T, so now, if the antisense says C, what's the sense side say?
G.
Now look at these two. Aren't they basically the same? If this one says G, he says, oh, look right there. If he says T, he says, well, look right there. What's going to be the only difference?
U.
Those U's. I can copy this again and just change the U's, the T's to U's, and I'll be done, but I'm not going to. I'm going to do it the old-fashioned way and line them all up. And then you'll see what we mean by the antisense. Now, if you look carefully and you compare these two, and you look at them, what's your only difference? Those T's and those U's.
Yep.
Yeah, and this is your messenger now. This is your messenger. Now what's going to happen, this thing here is going to actually go out to the cell. He's going to leave and be in the cell. Now when he leaves, this side will then just re-zip and go on living like nothing happened. I tell you right now that even though the messenger's out here, I can see it there, can't I? All I gotta do is change T's to U's, and I got it again done. So the messenger's gone. Now somewhere out here in the cytoplasm, there's a thing that looks like this. That should be big, and that should be small. This is a ribosome. It's a two-structure thing. All right? Let me erase that. Now, remember this thing that we just made? It's a U and an A and a G and a C and an A. I'm trying to copy and write. And a U and a G and a U. Missed that. Got it. And an A. This is out there in cytoplasm. He just left, remember? And if you look carefully, this series equals that series except for T's and U's swapped. Now, this thing right here is going to come along and insert himself right in there. And like you do in Braille, this thing will read this. And every three of these will tell him something. Now, when he sees these three, he flips them right back to where their words start. So when he sees a U, he flips it back to a what? An A. Which is what he wanted. And when he sees an A, he flips it back to a what? And he sees a C, he flips it back to a. Now if you look carefully at this codon and this codon, they are the same. He got the word opposite. Like if you're the messenger and say you're the you're the transfer. If I say up, what's opposite of up?
Down.
You say down. You say down, what do you do? Up, which is what I wanted. If I say black, you'll say, and then you'll say up.
Black.
Okay, black, which is what I wanted. And if I say true, you'll say, and then you'll come back with, it's kind of weird how it does it. It uses a lie to make it work, but it works beautifully because this thing, here's the antisense, here's the sense, and there's the anti, and basically this lie turns it right back to what I wanted. That's beautiful. That's binary code. It's as simple as that, binary code. And the body's been doing it longer than we have. I don't know if the folks that made computers saw this trend or not, but it's just back and forth. All it does. And like I said, you got to remember now, folks who are not trained won't know that this and this are the same thing. A, T, C. It's the same as A, because remember, this thing here, really, those are the same. So, by flipping it back, I have just gotten what the DNA said to have. So he'll get this one, and the transfer will come in, and you'll have A, U, C. And here's the protein, the amino acid, that was desired. And if you look carefully, this matches to that. And this amino acid turns out to be the very one that I wanted. And then the ribosome, this thing comes in here, and there's your acid. Now, this will leave with the acid gone. He'll leave. And now I'm looking for one like this one. Okay? CAU will give me what anticodon? CAU would give me what? Let me get my pen back. Okay. C will give me a what?
G.
G.
A will come back with a
U.
U. And U comes back with a
A.
A. Now look at this one.
Almost.
And look at this one. They are the same, except for the T. They're the same anyway. GTA is equal to GUA. Now, somewhere out here, there's an anticodon, there's a transfer, and he is carrying this message. And he will come up and match to this thing here. So, there's your next amino acid. And when this thing hooks right here, then this acid is hooked to that acid. And before long, you have a chain of acids that's been manufactured as you brought in one transfer at a time. Now, I got 3 good videos that can show you what I try to show you there. After seeing this mess, I think you'll comprehend better what the video is showing you now. So let's see the videos. Let's say that. This is. I would